Hello. Tēnā koutou katoa. I'm Joe Monks. I'm the president of the Society for Research on Amphibians and Reptiles in New Zealand and a member of the local organising committee. And it's my pleasure and privilege to introduce Dr. Geoff While, our plenary speaker for today, and he'll be talking about his research into the evolutionary ecology of family living in lizards. Thank you, Geoff.
Hello everybody, thanks Joe, and also thanks so much to Phil and the committee for inviting me. It really was a very unexpected and humbling invitation to receive, but I'm obviously very grateful for the opportunity to come and talk in front of you all today. So what I want to talk about today is to give you a little bit of an overview of some of the research that we've been doing, looking into the evolutionary ecology of family living, using one particular group of lizards, the Australian lizard group, the Evolutionary group. And then I want to use the kind of research that we've done and the kind of insights that we've gained, as well as kind of nod to the parallel work that's been doing in other lizard systems to really kind of spruik lizards as excellent model systems for trying to address some fundamental questions about how social complexity kind of evolves and diversifies. But before I do that, I'd like to acknowledge a whole range of different people that have been fundamental to the research that I'm going to talk about today. These involved obviously the fantastic collaborators that I've worked with across the years that have really helped shape the way I think about how family life evolves and how we can use lizards to address questions about how family life evolves. This includes a lot of people that are in the room today, Dave, Mike, Shai, Dan, Julia, Eric of course, Martin and Camilla, as well as a bunch of people that couldn't make it, like Tobias and Natalie. I'd also like to acknowledge a bunch of funding agencies that have supported this research. And of course, the wonderful PhD postdocs and honours students that have worked on these systems over the years and are responsible for lots of the research and data that I'm going to present today. Okay, so social behaviour is a relatively ubiquitous component of animal life. All animals, or at least most animals, are social to some degree. But we don't have to look very far to realise that social behaviour is also extremely variable. It varies within individuals, between populations and between species. In some systems, social behaviour extends simply to short-term interactions solely for the purpose of breeding. But in other systems, social behaviour extends to individuals reliably and repeatedly interacting with one another, resulting in the formation of stable social aggregations. Now, these stable social aggregations can either emerge because of the non-random associations between unrelated individuals, so basically unrelated individuals being brought together, as is in the case of these many egalitarian societies, but they can also come about through basically keeping related individuals together, as we see in these kin-based fraternal societies. Now, independent of whether these stable social associations emerge from an egalitarian root or a fraternal root, they can form the foundation from which a whole range of more complex forms of social behaviour can emerge. Things like cooperation, things where individuals forego their own reproduction to assist the reproduction of others, and ultimately systems where individuals become mutually dependent on one another. In extreme cases, these forms of complex societies have been identified as a new level of individuality, a new level of organismal complexity, and thus represent a major evolutionary transition. It's not surprising then that evolutionary biologists have spent a lot of time over the last decades and centuries trying to understand how these stable social associations come together, how they're maintained, and how they set the foundation from which these far more complex forms of cooperation and social behaviour can evolve. Okay, so usually when I get to this part of the talk, I look into the audience and I can see them thinking, this is all very good, this information about stable social organisations, transitions in organismal complexity is all very interesting, but I'm pretty sure you said you were going to talk about lizards, and I'm almost 100% sure that is a lizard. It could be a sausage on a log, but it's probably a lizard. And this is, of course, because lizards have always been viewed as relatively asocial creatures. That's not that they haven't contributed significantly to our understanding of social behaviour. They have. They've contributed to our understanding of communication, of sexual selection, of territoriality, of competition. It's just that they haven't really been thought to exhibit some of the more kind of complex forms of social behaviour that we say in, say, birds and mammals. Now, I think that this next slide kind of nicely articulates that. Now, this was something that was sent to me a while back. It's a list of books that was put together by an online bookstore called Abe Books. And they wanted to compile what they thought were the 100 weirdest book titles of all time. And those of you with a very keen eye can already see that there's this very sensible and very serious and very good book edited by Stanley Fox and others, right bang smack in the middle there. And I think this nicely articulates the position that lizards have in the public consciousness in terms of social creatures. So weird is the idea that lizards display social behaviour that the proprietors of Abe Books thought that it warranted position in this list. And I'm not sure what's more worrying about this, the fact that it's on this list at all, or that it seems to have sold less copies than lumberjack songs with yodel arrangements, a do-it-yourself guide to holy housewifery, or the radiation recipe handbook. I'm still waiting for my copy of Anyone Can Be Cool But Awesome Takes Practice. To be fair to the proprietors of Abe Books, it's just not the general public that had this view that lizards are relatively asocial creatures, or at least have a relatively limited capacity to contribute to our understanding complex sociality. This is something that also pervaded academic circles. And the best way that I could think about to show you this is to trawl through the various paper, fellowship and grant rejections that I've had over the years that seem to be based solely on this particular topic. Now, this is just the tip of a very large iceberg, but they nevertheless does kind of provide you with a little bit of an insight. My favourite one is this one in the middle. This is from an ARC, an Australian Research Council Discovery Grant. And the reviewer said, the main problem with this project lies in the choice of study system. If one wishes to address this question regarding the evolution of kin-based sociality in the best way possible, then I think most other vertebrate groups offer far better opportunities. Lizards are the last group I would use. Fortunately, this kind of idea has started to kind of disappear, and it's kind of disappearing under a weight of empirical evidence from a whole range of researchers working across the globe on a whole range of independent lizard systems, and they've started to show instances where lizards do indeed appear to live in more stable social associations equivalent to those that we might expect in, say, birds and mammals. Not only that, they've started to study some of the kind of consequences of this social life for kind of ongoing behavioural, morphological, physiological evolution. Probably the most well-studied group in this context is the Australasian Agurnia group. This is a group of about 6 genera and about 50 to 60 different species. Work into social organisation and family life in the Agurnia group was really kicked off by the late great Mike Bull and his work on the sleepy lizards. And Mike spent a lot of time in the 80s and since the 80s right up until his recent passing working on sleepy lizards, driving the roads out in Bundibor in South Australia, collecting information on male-female distribution. And what he found in the early 80s and the mid-80s was that he regularly found the same 2 individuals together, not just within a mating season, but between mating seasons. We now know that the record pair bond length for a sleepy lizard is 30 years, or about 30 years, 27 years. So really strong mate fidelity in this species. Mike's work on the sleepy lizards really kind of spurned an explosion of research into the Agurnia system and an attempt to kind of document the diversity in social organisation that we see in this group. We now know that the Agurnia group spans, species that are largely kind of solitary, through species like the sleepy lizard that live in pair bonds, right up to species like these Cunningham skink that live in large communal family groups with multiple cohorts of young and sometimes non-breeding adults hanging out within the social group, within their parent social group. So most of our work in this context is focused on this particular species. This is Lyophilus whitei, white skink. And I want to spend a little bit of time during the early part of this talk just giving a little bit of insight into what family life looks like in these lizards. Our work in this system is based on 2 key experimental resources. The first one is a long-term field study. This we carry out on the east coast of Tasmania in a place called Orford. It's a patch of open sclerophyll forest and it contains about 100 to 150 lizards. And each year we go out excitedly, fish for all our lizards, recatch lizards, remark them, remeasure them, and then we spend the year wandering around collecting positional data and also data on kind of any social interactions that we see. We've been doing this now for 15 years and we have data on about 300 adults that have been caught or recaught and over 700 offspring that have been born in the lab. The other facility that we have quite close to the university is this large-scale experimental facility, and these are 20 8 by 8 metre enclosures, or 10 16 by 8 metre enclosures if you want. And this is a really important facility because it allows us to kind of go into an APPOP system. It allows us to look at the variation we see in various social traits, derive hypotheses for the various environmental factors that we think might be driving variation, and test the causality of those associations in our experimental facility. And we've used this extensively over the past 5 or 6 years, and hopefully I'll have some time today to show you the power of this system for testing some of these ideas. Okay, so what does social life look like in this system? So once we get all our positional data, this allows us to create home ranges for each adult in our population, and we use home range overlap to give us an idea of the social structure and the pair bonding that's going on. This is kind of an example of what that looks like. This is our study site here. We have a kind of highway on the side. Well, it's a Tasmanian highway, so it's probably a couple of lanes to most people. And this kind of acts as a nice barrier to dispersal. We also have some unsuitable habitat down here that also acts as somewhat of a barrier as well. So it's a semi-closed population, but there is emigration and immigration. We derive home ranges for all of our individuals, and it looks something like this. And then, as I said, we use this to get some information about what social structure looks like. What you can see from this, well, you probably can't tell much from that because it looks extremely messy. But if we move or reduce this down to the 50% core area, then you start to see some patterns emerging. And this is where you get single black dot and single white dot occurring at multiple locations across the study site. And this is evidence of a single male and a single female co-occurring throughout the duration of the season. So not just in the mating season, but throughout the whole year. And what's important is that these black dots and these white dots, this core area, home range, sits on a really kind of important component of Agonia ecology. And that's their reliance on these complex crevice sites that they either construct themselves or they find kind of aspects of the habitat that they can utilise. So basically these single white dots and these single black dots are a male and a female sharing a burrow system together throughout the duration of the year. Most of these instances you find a single male and a single female, which is suggestive of social monogamy. However, in various parts of the sites, like here, down here, you find a slightly different pattern emerging, and that's where a male is able to either acquire a burrow that can accommodate multiple females, or he can acquire multiple burrows and thus multiple females. And these males are socially polygynous, and that accounts on average across years of about 30% of our social groups. These social groups are not just stable within years, they're also stable between years. So if you go to the same burrow system the next year, you're going to find the same two individuals together. We know that these guys aren't as long-lived as the sleepy lizards, but our maximum pair duration is still pretty impressive. It's 10 years and counting. And in fact, we actually find really low levels of divorce. So we very rarely find instances where you'll come into the population the next year and a male and female will have become separated. In fact, it only accounts for about 20% of all the pairings, which I think if you compare it to the pair separations of most Western human societies, it's a pretty impressive level of pair stability. Now, of course, we don't just want to know about the social structure in these lizards, we also want to understand a little bit about how that corresponds with the mating system, and also how variation in social organisation influences male and female reproductive success. So to do that, we jump back out into the NatPop about this time of year, and we catch all our pregnant females. We bring them into the maternity wards at UTAS, and we let females give birth. And this gives us information on female reproductive output. It gives us information on offspring, various offspring traits. But most importantly, it gives us access to a little bit of DNA that we can run up to the molecular lab genotype, and we can carry out paternity analyses. So what does that look like? Well, we know that about 30% of females in a given year don't reproduce. So females appear to skip reproduction every 3 or 4 years. Those females that do reproduce, we find that about 70% of those females are genetically monogamous. So they sire offspring only with their social partner. But of course, this leaves about 30% of offspring which are actually, on average, which are actually sired by males from outside the social partner. So these are extra pair offspring. Those fathers that sire those offspring tend to come from neighbouring territories. So this male sired a offspring in this female's clutch. This male gained additional extra pair paternity, and these two females clutches, these males kind of exchange paternity, and I don't really want to know what went on there. Some kind of party, keys in a bowl, and they let the good times roll. So obviously we're interested in how variation in these social strategies influence male and female reproductive success. We know that variation in both social pairing but also extra pair pairing mediates male reproductive success. So males can increase their reproductive success both by acquiring additional social partners and also by acquiring extra pair partners. So overall there's a positive Bateman gradient, so males can gain increased reproductive success by increasing the number of genetic partners he can acquire. We also know that females can gain, particularly from extra pair paternity, because this allows them to trade up from their social partner. So females that undertake extra pair copulations tend to do so with extra pair partners that are significantly less related to them than their social partner. And we know by comparing within an extra pair siblings that this has positive benefits for offspring heterozygosity. Now, of course, family life is not just all about the parents, it's all about the offspring as well. And in the Agurnia we have extended parent-offspring associations. This is what that looks like. So once offspring have been born, we go back out immediately and release them back out into the little burrows with mum. We let them run around for the autumn and we let them hibernate over the winter. And then we go and recapture them the following spring. And we look who's survived and also where they've ended up. This is kind of what that looks like for one particular season. We had about 40%, I guess 30 or 40% survival in this particular season. The red dots are instances of where we've found offspring outside the parental home range. So they've dispersed away from mum and dad's home range and they've found like a little crevice of their own, somewhere in the periphery of their parents' home range or somewhere else altogether. The green dots, however, are individuals that we go back the next season, we find them hanging out with mum and dad in their mum and dad's burrow. And they'll stay there usually for the rest of that, or the next 6 months until the next cohort are born, or occasionally they'll stay there for multiple kind of seasons. So we have prolonged parent-offspring associations in this system. This obviously raises an interesting question of what does this represent, some kind of primitive form of parental care? Are there any benefits to the offspring of hanging out with mum and dad? And the answer to that is yes. We've got a bunch of evidence to suggest that that offspring benefit from this association. First and foremost, the presence of a parent seems to almost completely eliminate aggression from conspecifics towards their offspring. This is fantastic work done by Dave O'Connor and Rick, not on Agurnia Lyophilus whitei, but on Saxatilis, the black rock skink, and they simply looked at how conspecifics responded to an offspring if in the presence versus the absence of its mother. When mum wasn't there, the offspring were likely to be attacked, and we know in the field they're likely to be eaten. But you put mum there and that almost completely eliminates this aggression. Mum doesn't have to be doing anything, she just has to be there. Kind of in line with that, we know both from the Saxatilis and also our own work on the whitei that female aggression tends to ramp up during gestation and is maintained in periods of postpartum care. So females become more and more aggressive as you go out throughout gestation, and then females retain that aggressiveness during periods of time when you might expect offspring to be around and also most vulnerable. This is one of my very favourite results that we've had over the last few years. This is work done by a PhD student, Kirk Munch, in collaboration with Dan. And Kirk was just interested in what the offspring did, what the presence of parents actually did to the offspring in terms of potentially eliciting some form of behavioural plasticity. So Kirk just kept offspring within and without mum and looked at how they behaved. And she showed the offspring behaved in a whole lot of different ways when they were with mum versus not with mum. They were more bold, so they were more likely to emerge after a predatory threat. They were more active and they were more exploratory. Again, mum's not doing anything, but the presence of mum seems to elicit this plastic response. And this, of course, might be quite important during the early stages of parental care evolution, if it provides these offspring with an opportunity to access resources that they might not be able to. And this kind of is in line with a bunch of work that Julia and Martin and others have been doing, looking at the role that kind of early social environment plays in mediating kind of behavioural plasticity. Following on from this, we know from our work both in our NatPop site but also work out at our outdoor enclosures that associating with parents, particularly mothers, increases offspring growth across the first sort of 6 months of life. And finally, you know, the flip side to parental care, as any of us that are parents know, is that parental care is not just beneficial for offspring, it's also damn costly to the parents. But not if you're a Gurnia lizard, because we've never really been able to find any costs associated with care in these guys, not in terms of survival, growth, harassment, future reproductive success. And again, this might be quite important during the early kind of evolution of parental care. The parents play a relatively small cost to actually tolerating these offspring. Okay, so hopefully that shows you that we have family life in these guys, and this family life is underpinned primarily by long-term stable pair bond and delayed juvenile dispersal. And this is kind of the central tenet of family life, not just in our whitey eye, but right across the Agurnia group. So if we look at all the species that have been studied in this context, they all seem to have these two kind of key features, except for some of these solitary species like Liophilus ornata. But of course there's huge variation. You know, you get up to, as I said, Agurnia cunninghamia, which don't just tolerate one cohort, but tolerate multiple cohorts of young. So we have these kind of key traits here that are kind of the central tenets of family life. But, you know, getting to this point is relatively unsatisfactory, particularly if you're an evolutionary biologist, because we don't just want to identify pattern, we also want to know something about process. We want to know what factors have actually influenced the evolution, the emergence and evolution of these forms of family life. And much of our work over the last 5 years is trying to use this system to do exactly that. And lizards are really neat for this because their family life really is very, very simple. And what you can really view it as is just changes in how individuals associate with one another. And so that allows us to ask a whole heap of really neat questions. It allows us to ask questions about the origin. How do you get these entities together? How do you get males and females together for a prolonged period of time? How do you get parents and offspring together for a prolonged period of time? It allows us to ask questions about how those associations are maintained once they first emerge. And finally, once these associations are stabilised, it allows us to ask questions about how the stabilisation of those associations sets the foundation from which you can get the elaboration of social behaviour and social complexity. So to start off with, we've done a little bit of work on the origin. So this is all about trying to ask how you get these associations in the first place. We've focused primarily on associations between parents and offspring in this context. How do you get prolonged associations between parents and offspring? Like work from birds and mammals will tell us that life history traits are going to be important in this context, and ecology is going to be important in this context. Life history traits is basically going to set the stage for which ecology can act. One life history trait that we've been particularly interested in this context is viviparity. And that's because we think viviparity is going to be really important for increasing the opportunity for ecology to act. So if you think about if you have an oviparous mum who lays an egg, and then there's some separation, temporal separation between when she lays that egg and when the egg hatches and the offspring emerge. Mum can go and do any number of things in that time. You know, there's a separation between the presence of a mum and when she lays the egg and where she is once the eggs hatch. That's not the case in viviparous species. Mums are always going to be present at the production of their offspring in viviparous species. And that means it potentially is an easier route for ecology to act on those associations in viviparous species versus oviparous species. So we wanted to test this idea. We can't do this in the Agurnia because they're all viviparous, so we kind of took a step backwards and looked at this right across lizards and snakes. And this was done with a PhD student of mine, Ben Halliwell, and he trawled the literature kind of based or expanding Lewis Somer's early work and collated all the information he could find on parent-offspring associations. So these aren't the same associations as we see at the Agurnia. They can be much looser, so they can be like associations that just go for 2 weeks or 3 weeks or several months or several years. He found evidence for parent-offspring associations in about 95 different lizard species, spread I think across about 20 different families. And what we wanted to do is understand how these associations fit with the known distribution of viviparity. So this is what that looks like. This is the whole squamate phylogeny. Viviparity in red here and oviparity in blue. And these black dots here are the instances where we see parent-offspring associations. You can probably already tell that these black dots tend to predominantly, but not always, occur on the red components of this circle. In fact, 66 of the 95 species that exhibit parent-offspring associations are viviparous, despite the fact that 80% of species are oviparous. So pretty nice initial evidence that viviparity might be playing a role. But of course we wanted to kind of look at this a little bit more formally. And the tough thing here is that it's difficult to assign, or it's easier to assign presence of care than it is to assign absence of care. So Ben set some very strict criteria that allowed him to also assign absence of care, which reduced our data set down to about 1,200 individuals, again, 95 of which had these parent-offspring associations. And this allowed Ben to do some sophisticated phylogenetically controlled comparative analysis to look at the correlated evolution between parity mode, viviparity and parent-offspring associations. And to cut a long story short, and what is fairly obvious from correlated phylogenies here, parent-offspring associations are far more likely to evolve from a background of viviparity than they are from a background of oviparity, suggesting that viviparity is potentially a really important precursor to parent-offspring associations in this system. Of course, it doesn't actually mean that you're going to get parent-offspring associations. It's only going to increase the likelihood. Ultimately, you need ecology to actually keep individuals together. In the Egernia, we have one particular ecological factor that we think is particularly important in this context, and that's this reliance on deep and complex burrow systems. These are burrow systems, they either sort of excavate themselves, or it's systems that they kind of utilise, like this massive lizard apartment here that's towering over Martin out in outback New South Wales. Now, it's much more difficult to get good comparative data on a reliance of crevice sites, so we can't really, or haven't been able to yet, test this in a comparative context. But what we can do is use the kind of facultative nature of the parent-offspring associations that we see in our YTI, and we can test experimentally whether restriction of habitat mediates parent-offspring associations in our YTI. And this is more work done by Ben, where we used our experimental facilities, and what we did is we used our double enclosures. So we've got a home enclosure here, a dispersal enclosure. They're split, but they have dispersal corridors, which we covered with fine mesh that only meant only offspring could move from one to the other. Then filled one side full of parents or mothers and their offspring, and then we manipulated the saturation of habitat on the other side. And we watched the offspring for 8 weeks, and we worked out what they did and where they ended up. And what we got was some really nice evidence to suggest that habitat saturation is indeed a kind of important factor mediating parent-offspring associations. So we didn't find any difference between the two treatments in which whether individuals explored, so you got equal exploration of the dispersal corridor, but we found significant differences in whether offspring settled in the dispersal corridor between the two treatments. Offspring were far more likely to settle in that dispersal corridor at the end of the experiment in the low treatment, where habitat wasn't saturated, versus the high. And this had consequences for the level of parent-offspring associations. We found almost twice as many parent-offspring associations in the high saturated treatment. So this suggests that habitat saturation is an important factor mediating parent-offspring associations in our system, and has probably been important in influencing the emergence of family life more broadly in the Egernia. Okay, so once you have ecological and life history traits that bring individuals together, you have individuals that are interacting with one another, and each of those individuals has its own idea about how it wants to maximise fitness, and that's going to influence or result in conflicts of interest about the best way to go about fitness. So we're going to get conflict, and for these systems to stabilise, we need to kind of moderate that conflict in some way, or that conflict needs to be moderated. You need to reduce conflict, increase cooperation. So what does conflict look like in these little lizard societies? This is just a little video of what happens soon after birth. It's a mother up here and 2 offspring. So pretty much exactly like my house, half an hour before bedtime. And what's really neat about this is we can quantify this conflict in various ways, and we can ask what kind of influences this conflict. When we actually have a look at where the conflict lies in these lizard families, we find lots of conflict between siblings, lots of conflict between fathers and offspring, but almost no conflict at all between mothers and offspring. Again, very similar to my family life. So if we want to kind of maintain, or we want to stabilise this system, ultimately we need to think about what are the kind of factors that are going to mediate conflict between these entities. Luckily, we have a pretty strong theoretical foundation for kind of understanding what moderates conflict, and that's Hamilton's rule. So we know that conflict is going to be moderated and cooperation enhanced based on the level of relatedness between individuals, and also the costs and benefits of interacting with one another. So any factors that mediate these terms are going to be important in moderating conflict. Relatedness is primarily mediated in these systems by female polyandry. If female's mate outside a social pair, she reduces, obviously, relatedness between a social pair and her offspring, and she potentially, if there's multiple paternity in the clutch, decreases relatedness between siblings. She turns full siblings into half siblings. Resource availability is likely to influence the costs and benefits of interacting between siblings or between parents and offspring. And there may also be some direct benefits of living in a group that may also mediate the stability of these systems. Now again, lizards are really great for kind of studying the relative roles of R and B and C in these systems, because we can manipulate female polyandry by bringing them to the lab and giving them access to different partners. We can manipulate resource availability. We can see how this influences conflicts and family dynamics at multiple kind of different levels. So we can manipulate them, and we can look at how this influences conflict at kind of the brood level. We can go out to our outdoor enclosures and we can manipulate these things and see how this mediates offspring dispersal and family dynamics. We can go into our natural population and we can see how temporal and spatial variation in polyandry and resource availability influence variation in family dynamics in our nat pop. And ultimately we can go up to our kind of between species systems and ask how levels of polyandry and resource availability might influence kind of broader macro-evolutionary patterns of family life. And these are things that we're all currently doing at the moment, in collaboration with Mike and Dave and Martin, Tobias and Charlie and Eric, trying to put all this together to try and really understand how R, B and C mediate levels of conflict and ultimately family dynamics. And hopefully by the time the next World Congress comes around, we can be able to talk a little bit more about that. So one little kind of aside that I did want to talk about is, I mentioned this, that there's kind of limited evidence that mum's getting involved in mediating any of this conflict. And that begs the question whether mum has alternative mechanisms which she can mediate this conflict. And the answer in this Agurnia system is actually, well, she does. And that's because the Agurnia kind of exhibit another weird kind of behavioural trait that we've termed birthing asynchrony. This is common in Lyophilus whitei, but it's also been observed in a number of other species. And this is where mum, she gestates all embryos together, so all embryos gestate synchronously, but when they're ready to be born, she gives birth to one offspring and then holds on to the other offspring. She can do this usually for about 2 days, but we set a new record last year where a female held on to an offspring for 15 days before giving birth to the next offspring. Now this, of course, creates a massive size hierarchy in the clutch, which shifts the competitive kind of environment within the clutch. Very, very analogous to what we see in respect to hatching asynchrony in birds. Now, we've spent an awful lot of time trying to understand why females are doing this, what kind of situations might females, because it gives the females potential to mediate levels of conflict within the brood simply by changing the spread between births. As I said, we've spent a lot of time trying to work out the kind of conditions by which females might facultatively alter their birth spread. And we haven't really found anything concrete yet, but we certainly are still looking. But potentially the really kind of interesting question that emerges out of this is not why they give, why they've kind of evolved this behaviour, but how the hell do they do this? So they can hold onto embryos not just between uteri, but within uteri. So they can give birth to one offspring within a uteri, hold on to the other one for several days. And this suggests that they've evolved somehow really kind of fine-scale control over the birthing process that has allowed them to do this. And in collaboration with Camilla and a PhD student of ours, James and Martin, we're kind of starting to try and understand how they actually achieve this. This involves, doing a bunch of kind of contraction assays where we can look at within and between uteri response to various birth hormones and how this might vary, and combining that with some molecular work looking at specific receptor genes and how they vary within and between uteri, and hopefully in the near future some kind of whole transcriptomic work. So again, hopefully we'll have a little bit more information on this by the time the next World Congress comes around. Okay, so you've got life history traits and ecology that bring individuals together, relatedness and costs and benefits that kind of keep them together, and this sets the stage from which you can get further diversification. So where ecological constraints are relaxed or levels of female polyandry go up and relatedness goes down, this could potentially result in selection for decreased investment in offspring and potentially the dissolution of kind of social systems. Alternatively, if ecological constraints become more severe, relatedness or monogamy goes up and you increase relatedness, this could actually potentially select for greater investment in offspring. For example, investment in offspring across cohorts, resulting in transitions to more complex and larger social complexity, like we see in the Stokesii through Mike Gardner's work and also the Cunningham skink, where you get multiple cohorts of offspring hanging out together. So we can use this to understand how you get transitions in social complexity from simple social groups to more complex social groups. But it also gives us an opportunity to kind of think about how these systems provide a foundation from which you can get selection of more complex social behaviour traits. And parental care is a really neat kind of system as far as this is concerned, because you can kind of think about how these association between parents and offspring sets the stage from which you can get more complex forms of parent-offspring interactions. And I just wanted to show this little video. This is something that we see relatively regularly or occasionally, I guess, and this is an interaction between a mum and an offspring. You can see the offspring there just chewing away on that little mealworm. Mum's not too happy, but you know, she's not super angry either. Just eat away for a little while. Mum quickly tries to eat it. No. Off it goes. Whoops. Now, I don't want to suggest for a minute that that's direct parental provisioning or anything like that, but the point I want to make is that once you get these associations between parents and offspring, and once you stabilise them, it sets the foundation from which you can get selection on a whole range of female traits to kind of function in this novel social context. So variation in female aggression or how females feed, you know, can then be co-opted and refined to function in this new social context, potentially providing the foundation for which you get the evolution of more complex forms of parental care. Okay, so to sum up, anyone who's kind of read anything about social evolution will kind of start to have recognised some pretty similar themes. We've got life history traits, we've got ecological constraints, we've got kin selection. And that's not really surprising, because these are the factors that have mediated the emergence of kin-based sociality across a wide range of organisms. Evolution is often convergent, and kin-based social organisations come about because ecology and life history traits act together to bring individuals together, and kin selection helps refine those associations. This is what happens across insects, across mammals, across birds, across fish. And lizards are no different in that respect. But what lizards give us that is beneficial over and above some of these other systems is a number of things. First of all, they're really amenable to experimental manipulation. This allows us to really kind of test causality of these different kind of environmental or life history factors, or R or BC, that might mediate aspects of social evolution. They also show huge variation in social complexity. This is particularly the case in the Agurnia. Now, we haven't yet found a eusocial Agurnia. I haven't given up hope that one day someone will be digging in the desert and they'll unearth a massive queen Agurnia with thousands of kids. But until that happens, I think we've got to be satisfied that we have the rest of the gamete of social complexity, from solitary species right up to species where individuals appear to forgo their own personal reproduction and hang out in the group, something akin to cooperative breeding. And this is really neat because we can take all the microevolutionary work that we're doing with our lap pop and also with experimental work, and we can ramp that up to kind of ask questions about how that links to macroevolutionary patterns of diversification, social diversification. So that leads to the question I quite often get asked about this. Okay, well, if these processes are so simple and so convergent, why don't we see more, why are there not more social lizards? And I think the answer to that is there are. We know that there are, but there are probably even more that we know than the ones that we know about. We just need to look. And there's fantastic research being done all around the world on various systems that have very analogous social systems to what we see in the Agurnia. Julia Riley's work on the cordylids, Alison Davis's work on xanthusids, work here by Steph Godfrey on the oligosomids, Yin Chi's work on the phynocephalus, and Stan Fox's work on the Liolaemus. All of these systems are based on the same kind of central tenets, or at least parent-offspring associations that we see in the Agurnia. All are live-bearing, all are long-lived, all rely on particular habitat constraints. What's really neat about these systems is that they represent evolutionary independent transitions to family life. And we don't get this in birds and mammals. So we've got 6 at least independent transitions. And this allows for an extremely powerful comparative context for us, questions about how these transitions have come about, how they've come about from a kind of phenotypic perspective, but also how they've come about kind of from a molecular and genomic perspective. And I think what that means is that hopefully in the next years and next decades that people will greatly appreciate much more the potential role that reptiles are going to be able to play in addressing that fundamental evolutionary question of when, where and why do animals live together. And so with that, I'd like to spruik the symposium tomorrow that's organised by Julia and colleagues that's going to kind of explore this topic a little bit more. Lots of fantastic talks from some of the other systems that we've talked about, also from Agurnia pioneers like Mike Gardner, who's going to give a talk as well. So if you're interested in this stuff, then please come along. Starts at 10 in Castle 2. And with that, I'll invite any questions.
Thank you very much, Geoff, for a fantastic talk. I really enjoyed it. Opening up for questions both here and in Castle.
Hi, that was great. You alluded to environmental constraints towards the end, so I had a couple of little thoughts on that one. When the young are dispersing or not dispersing, one might expect that there'd be a high degree of parent-offspring interactions in an environment with a lot more food. That was the first thought. The second thought was about birthing asynchronity and the idea. I don't know to what extent females can absorb the fetus, but one would think that if they were able to engage in a mechanism whereby they could delay birthing between births, that in a low-food environment they might have a much longer period, and that would result in a social system where there could be a lot more aggression between the offspring, i.e. the big ones might eat the very little ones that are resulting from this long interval.
Yeah,
Sorry about all that, but there you go.
Yeah, no, exactly. I mean, and these are some of the things that we are trying to manipulate. But you're right, like we've often thought about, particularly the birthing asynchrony as a mechanism, like in birds, where females can kind of alter the competitive environment during either resource poor or resource high years and kind of manipulate the amount of who's going to stay and how many offspring are going to stay. And we don't see many costs, obviously, to the females of having offspring stay around, but there may be kind of hidden costs that we don't know about. But there's certainly lots of potential for these subtle changes in resource availability to manipulate, to influence kind of levels of conflict, and also for female to then respond in various ways. Yeah.
There's a question up the back.
The video that you showed of the conflict between the two young lizards, it got me thinking about sort of my own interactions with my little brother.
Yeah.
So my question is, how would one distinguish between true antagonism versus play, and have we ever seen play in lizards?
Yeah, so I was talking to Gordon about this the other night, and of course, I mean, some of these relatively subtle kind of interactions, it's relatively difficult to kind of assign intent. What I will say is if you leave these siblings together for too long, they don't survive for too long. You know, so you get one sibling basically ending up, it's siblicide. And I remember playing with my brother, and he's still around, so I think that probably suggests that yeah.
Okay, time for one last question, thanks.
Hey, Geoff. Great talk, thanks. I was wondering about the phylogenetic correlations you were showing between viviparity and parental care, and sort of wondering about some of the causation, because one could argue viviparity is already a form of parental care, and so those systems might already be under selection for extended parental care that could then extend beyond birth, and they've also invested a lot of energy and resources into those young potentially.
Yeah, exactly. So I mean, my view of this is that, like, I think the kind of opportunity is a very parsimonious explanation, but there are lots of alternative explanations. One that we're kind of thinking about exploring soon is kind of the idea that potentially viviparity mediates things like kin recognition, so potentially prolonged association between offspring in utero might influence the recognition mechanisms between parents and offspring, and that be another route by which viviparity could influence the likelihood that you get parent-offspring associations. So yeah, there's a bunch of potentially alternative explanations to how viviparity and parent-offspring associations co-evolve.
Thank you so much, Geoff. If we'll join in clapping again, applauding again. Thank you very much. Thanks a lot. And Phil's got a little bit of housekeeping, but first just a message for members of the Society for Research on Amphibians and Reptiles in New Zealand. You'll know that we've got our AGM at lunchtime, and the program, it looks like it's the full hour from 12 to 1. We've clarified by email, but not everyone's reading emails. So the plan is that we're having a council meeting for all council members at 12:05. We'll approve those of you that wish to join at that council meeting so that you can come to the AGM at 12:15. Bring your lunch. Please try to be on time. Castle C. Thank you.
Great, thanks, Joe. Okay, so just a few housekeeping notes. The first one is about the Congress dinner. So the Congress dinner is on tonight, and in the programme, unfortunately, it says 6 o'clock. It actually starts at 7 p.m., so if you're not coming to the Congress dinner, just pass that message on to anybody who's not here anyway. So Congress dinner starts at 7 p.m. It's in the town hall, and you have to make your own way there. And what you can do is go across 2 blocks that way and then keep heading down until you hit the town hall. It's very easy to find. The other thing that I wanted to say is about the silent auction. So the silent auction closes today after afternoon tea. So please go along and try and bid for those items there. All the money raised at the silent auction will go to student scholarships to attend the World Congress number 10. So we had an AGM last night, and the people who were successful in the auction will need to come and collect their goods and pay on Friday. So we had the AGM last night. Many of you were there. You voted on your next executive committee. You also voted on where you would like to see the World Congress number 10, and that was done on the app. Many people were concerned that they were voting twice, but we've actually found out that it's your last vote that counts. So if you did vote more than twice, then it will be the last one you voted for that will actually get counted. So that was that. Something I wanted to say, we also had a declaration. So as the World Congress of Herpetology, of which you are all members because you're delegates at this conference, we've made a declaration about climate change and amphibians and reptiles and how important that will be. That we will put up on the app, and the declaration will come out from this meeting as a ratified declaration from all of the members that were here. I just wanted to say a quick word about this. Many of you will know Tim Halliday. Unfortunately, Tim died last year, and in honour of Tim, we are carrying on his legacy. He used to collect as many as he could papers about amphibian conservation and lodge that on Amphibia Web. Tim was not particularly strong in his computer skills, so he used to do all this manually and search for all these papers manually. Now with the advent of computer skills, we've employed a research assistant based here at the University of Otago, and we try to collect every paper there is on amphibian conservation, and we publish the lists of those, and often with links to the papers. So if you've got any students in amphibian conservation or you're working in amphibian conservation, it's housed in 2 places. It's housed on the Amphibian Web, and it's also housed on the IUCN ASG web page as well. We've named it in honour of Tim. It's called the Halliday Conservation Library, and it should have every paper there.